Thanks for coming, everyone. Wow. Okay. Are we ready? This is the last. Okay. So this is going to be short and sweet because I do feel for everyone who's worrying about this lecture for tomorrow morning. So we're going to just try to get some main points of apoptosis in. I didn't want to let you go without teaching you apoptosis. So sorry, here you are. Tomorrow morning.
Wait, what?
Tomorrow morning. Tomorrow morning. Here. Don't miss it. I don't do makeup. Okay. So far, this is what we've done with cell cycle. So if you study this and really understand it, you should be fine. Right? We talked about the different methods. Really understand your methods there because I'll ask you to use them. The different phases of checkpoints, regulation of the cell cycle by CDKs, and how it's controlled by cyclins, different ways of yes?
Even if you don't put up the lecture, can you put up the PowerPoint?
PowerPoint's already up.
Oh.
Yeah, it's not that the lecture won't be up, it's that by the time I get to it, because I have to leave here, pick up a child, cook dinner, and get them in bed. Yeah, I only pick up one child. The other two are somewhere else. They're all the way across town. Dad gets them. Anyways, that's my routine. Okay, so the lecture will get up, but it might not be till midnight, and I don't want you sitting there cursing my name. Though you might, anyways. Okay, so regulation of S phase by APC and CDK, specifically really understanding why replication happens only once per cell cycle. Understand MCDK and how that regulates mitosis and mitotic spindle assembly. Understand APC. APC does a lot. We talk about it again and again, and all the things that get degraded to turn things off. Understand APC and mitosis, how you exit mitosis, what APC degrades in mitosis. Understand your spindle assembly checkpoint and how APC is inhibited by unattached kinetochores. Then, of course, we did cytokinesis, and we were talking about actin and myosin regulation. So, again, bringing in the cytoskeleton to this whole thing. Again, information of a contractile ring, and then keeping CDK activity low at G1, how that's accomplished. How mitogens stimulate cell growth, how DNA damage causes cell cycle arrest, and then how excessive cell proliferation triggers cell cycle arrest. And we went through a bunch of mechanisms, so really understand it. Don't just understand DNA damage causes cell cycle arrest, because then you've learned nothing. Understand how it happens. Okay, so apoptosis. We'll start it with a movie with no volume. Oh, and a dog. Aww. That's what happens. Skip that.
Apoptosis.
Okay, so this is looking at cells. You know, these are the individual nuclei, and you're going to see they all start kind of imploding, kind of, and they bleb. It's called blebbing, which is like making these, and you can watch it on your own computer. You might get better resolution. But you can see each one. There's one blebbing, there's one blebbing, there's one blebbing. So they're going from this nice shape. And so this is a close-up here.
We observe the process at higher magnification.
And you can start to see, oh, there you go. And then there's the blebbing. And so it's kind of packaging its contents within membrane. Yeah. I'm not sure what they did in this exact experiment and process that made it light up a little. I don't know that it would naturally fluoresce, yeah. And so there you can really see, there's your blebbing. You can see them kind of blebbing, that membrane kind of blebbing out. It's nice because then you don't like have cell parts all over you, right? Because then you could generate an immune response to the inside of your cell. So it's kind of nice and neat. cell death. Thank you, Kyoto University. So you guys can watch that, tonight when you're trying to study or calculating your grade. I know that's what you guys do. Okay. So some main points. What I really want you to get out of this lecture is what is apoptosis in terms of what regulates it, and that there is an intrinsic and extrinsic form of apoptosis. So you're not going to get a so far slide for this. So just really, I'll kind of try to point out things you should focus on. So here, this is just telling you that apoptosis depends on activated caspases. And a caspase is an enzyme, and the reason it's called a caspase is because it has a cysteine in its active site and an aspartic acid. So the aspartic acid is actually where the enzyme cleaves on its target protein. So that's where you get your CASP, caspase enzyme. So you can try to use that to remember. It has a cysteine in its active site. Well, most proteins have cysteines in their active site. I'm not going to test you on that, but it does kind of help to make you remember it's an enzyme and it cleaves target proteins at aspartic acids. Okay, and so remember caspases are these enzymes that get activated and they cleave all sorts of things in the cell, and that's what induces apoptosis. So you normally have these inactive procaspases, and then you get cleavage, and they kind of change their conformation and become an active caspase, and then they can go and cleave other things. I just want you to know not all caspases are involved with apoptosis. Some are involved with inflammation, just so you know, because at some point you might come across a caspase that has nothing to do with apoptosis. And then is this cascade where a caspase is activated and it cleaves another caspase, and then those caspases go and cleave all these different cellular proteins, and then boom, apoptosis. So you can think of caspases as part of two different categories. I like how the book does this because they're really numbered, and sometimes when there's caspase with a lot of numbers, it's hard to keep track of what's what. But think of it as there are initiator caspases and there are executioner caspases. And an initiator caspase is just going to activate the executioner caspases. And then the executioner caspase is going to cleave the proteins in the cell. And so you'll have one active initiator caspase, and it'll cleave and activate many executioner caspases. And then those caspases will go on and cleave cytosolic proteins and nuclear lamins and all sorts of things, and then you have apoptosis. So really the most important thing is this first activation. Okay? So you might say, okay, well, what are these caspases cleaving? Well, there's all sorts of things. You've got cytosolic endonuclease inhibitors. Yes?
So you said that the initiator starts the caspase cleavage, and then the executioner,
It cleaves the targets.
Oh.
Yeah. So just think of it as there's something to turn it on, the initiator. So actually you can remember that because it's initiate.
Right.
And then executioner is the actual executioner. Scary. Cleave things and so on. Okay. Are we done? No. Okay. So targets of these caspases are the cytosolic endonuclease inhibitors. If you think about what that is, an endonuclease inhibitor would be what? What would inhibit? Other than endonuclease. What does an endonuclease inhibitor inhibit? What process? What's an endonuclease? When did I learn these things? I don't know. Maybe it just came to me in the night. What's nuclease? How's that? Break it down. Enzyme cleaves what? nucleotides, right? Because nucleotides, right? So an endonuclease is an enzyme that cleaves DNA, RNA, depending on which nuclease it is. And so these caspases are going to cleave endonuclease inhibitors. So now endonucleases can't be inhibited anymore. What's one of the major things that happens in apoptosis is cleavage of the DNA. And that's because all these endonucleases are not inhibited anymore. Nuclear lamins, that's involved with the nuclear structure, so it kind of cleaves apart the nuclear envelope. So now you have DNA all over inside the cell, and it's able to be cleaved by all these endonucleases. It cleaves cytoskeletal proteins, right? So cytoskeletal proteins such as actin, microtubules, these are really important in cell structure, so that's why you see the cells rounding up. It cleaves adhesion molecules. These are cell surface proteins that are involved with attaching cells to wherever they are, whatever surface or neighboring cell. So all of these are cleaved by these executioner caspases. And so you can see how that might result in cell death, right? I'm so funny to myself. Okay, so two pathways for apoptosis. There's your extrinsic pathway and your intrinsic. Extrinsic really just means your stimulus that's causing apoptosis is coming from inside or outside of the cell? Outside, because that's what extrinsic means, right? I just need. Thank you. So here's an example of an extrinsic pathway. It really has to do with a receptor and a ligand. So remember we learned about all these different types of receptors. Well, they're also death receptors. And death receptors, when they bind to their ligand, induce apoptosis. And so here this is just showing, this is in the immune response where you'll have a killer T cell and you've got some infected cell, let's say, and its job is to cause that cell to die, because usually you want to kill infected cells if you're a T cell. And so it has, there's FAST, which is the death receptor, and FAST ligand, which is another receptor on the killer T cell. And as soon as that death receptor is bound, there's all these different proteins that all are recruited to the death receptor, which form this disc complex. And the whole purpose of that happening is to activate the initiator caspase. In this case, it's caspase 8 or 10. And then that initiator caspase can go activate the executioner caspases, and then you have the cleavage of all those things we just talked about, and then you have cell death. Okay, that's apoptosis. What's very cool is viruses. They have decoys for a lot of these things. And so you can end up with these procaspase decoys, so you'll get activation of, you won't really, you'll like flood, the virus will flood the cells with caspases that really don't get activated and don't truly do anything, and can inhibit apoptosis that way. So that's the extrinsic. So your stimulus for apoptosis is coming from outside the cell through ligation of a death receptor. So just really understand. So this topic is really nicely, easily organized, because you can think of it as, you know, apoptosis, major concepts, caspases, initiator, executioner, and then get more specific where you've got your extrinsic and your intrinsic pathways. Okay, so here's your intrinsic pathway. So here your stimulus is coming from within the cell. It might be from DNA damage, hypoxia, stress, lack of some sort of extracellular signals, survival signals. And so here, this involves the mitochondria and all that cytochrome C that's in that intermembrane space. So here you'll have you'll have release of cytochrome C resulting from the apoptotic stimulus. And then you'll get assembly of this apoptosome. And the cytochrome C actually forms part of the structure of this apoptosome. And the purpose of the apoptosome is, again, to activate that initiator caspase. So it's all about activating the initiator caspase, which triggers the whole thing. And then you get activation of executioner caspases and then boom, apoptosis. And so the cytochrome C plays a role. It actually is involved in the structure of the apoptosome. If you don't have cytochrome C, you can't get assembly of this apoptosome. You can't get cleavage of your procaspase, and then you can't get activation of apoptosis. Okay? How's this going? Is everyone enjoying this? I'm so glad you all came. I should have brought candy. I wish I had candy. Yeah, I went to the departmental, no, it was a college holiday party today.
I heard about those.
Huh?
Was it in York?
Yes.
I think I walked by.
Yeah. Yeah. And there was no food left. No, so I walked all the way over there. There's no food. Hey, once a poor graduate student, always a poor graduate student. So I was like, free food.
I know, always is something.
I got a soda. I got free soda. Yeah, I'm wild. They did have punch that had Southern Comfort in it, and I thought to myself, and then I thought, oh wait, I have to teach. So that would have been, I don't think it would have been that different. It would not have been that different. I probably would have just been snoozing a little bit. Like I would have been more like this. Because that's what happens when you have 3 kids and you drink, you go to sleep. Okay. Okay. So this is how we study apoptosis. So this is a kind of important thing to think about. So if you're studying apoptosis, you need to find a way to actually say, oh, this has induced apoptosis. So there's 2 things. One is, all this cleavage activates these nucleases that result in cleavage of DNA. And so if you take the DNA from the cell, over time, after inducing apoptosis, you'll get this characteristic ladder band. And that's because when your DNA is not cleaved, it's one gigantic thing. And when you've got nucleases chopping it up, you get lots of different sized pieces of DNA. So this is very small, this is very big, and so that's why you get this ladder. Okay? So if I gave you a question where I said something like, blah, 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 treatment, blah, 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 you get this, you should be able to tell me whether or not apoptosis occurred and what time, what time did it occur, at 0 or 12, right? Because you're only going to get that cleavage after apoptosis happens, okay? Do you guys understand how we're seeing cleavage here? Because if you've got DNA that's like thousands and thousands of base pairs long, it's going to migrate very slowly through this agarose gel, and it's going to be at the top. But if you've got where it's been cut, it's randomly cut. It's not all cut in equal lengths. And so you might have something that's 100 base pairs, something that's 500 base pairs, something that's 1,000 base pairs, and then it all kind of line up in this ladder. That's why it looks that way. Yeah.
I'm sorry, I'm confused. So during apoptosis, I thought that we were inhibiting the endonuclease.
No, it's
Or are
cleaving
we
the
inhibiting?
inhibitor. Here, let me go back to that.
Okay.
Targets of executioner caspases, cytosolic endonuclease inhibitor. So
We're inhibiting the
you're
inhibitor.
inhibiting the inhibitor.
Okay.
Yes. So the question was, wait a minute, I thought we were inhibiting endonucleases. No, you're inhibiting the inhibitor, which is actually kind of active. You're liberating the endonucleases and allowing them to become active. Okay. So another way to study is doing it. Yes.
Okay, so the caspase and the extrinsic pathways are 8 and 10.
Huh?
The caspase and the extrinsic pathways are 8 and 10.
8 and 10, but I'm not going to ask you these details. just understand initiator and executioner. I mean, you can memorize it. But I mean, save your time. But yeah, so in 8 and 10 are your initiators in extrinsic. And in intrinsic, 9 is your initiator. Yeah.
Can you say that one more time?
Extrinsic, you've got 8 and 10, which are your initiator cast bases, and intrinsic cast base 9. I promise if I give you a number cast base, I will mention whether it's an initiator or not. I promise. And if I don't, just come up to me and say, you promise. Okay, good? Okay. So another way to see whether or not apoptosis has happened is a tunnel assay, and it detects DNA ends. So again, you're still looking at cleavage. You're getting lots of cleavage of DNA. You're going to have lots of DNA ends. Normally, you wouldn't have many DNA ends, right? How many DNA ends do you have on a chromosome? So you.
Good question. That was a great face. I was like,
What if you're bacteria?
none.
Thank you. For a tunnel assay, it detects DNA ends. So when you have a lot of them, you can, I forget, it's an enzyme that transfers something to the end, and then you can see color. And so when you get, so this is actually looking at the development of frog feet. Well, either way, any feet or hands. The way, they start out like this, and then you get apoptosis in between, and that's how you get your individual fingers or toes or whatever. And so here, they're showing you get this positive tunnel in this area where all those cells are going through apoptosis. So a tunnel assay is another method for detecting apoptosis. So if you have a positive tunnel assay, that means there are a lot of DNA ends. Apoptosis has occurred. If you don't have that many ends, you shouldn't really, if there's only 2 ends to detect, you're not going to see much. Okay. So BCL2, so we did extrinsic, so this is kind of continuing on with intrinsic. The BCL2 family of proteins regulates the intrinsic pathway of apoptosis. So they can be pro or anti-apoptotic depending on what they do. They're a BCL2 family of proteins, which means they share similar domains. And this is just saying, in reality, they all share this BH3 domain. Okay. So here's your intrinsic mechanism. So basically, there are these pro-apoptotic BH1, 2, 3 proteins, and they form a pore. So there's some kind of stimulus, and they form a pore in the mitochondria outer membrane, and that releases all these different proteins. Cytochrome C is one of them, which is why you see cytochrome C release when you have apoptosis. So this is called mitochondrial outer membrane permeabilization. You get release of cytochrome C from the intermembrane space. Okay, cold calls, cytochrome C comes out. Remember, what was cytochrome C doing? It was helping to form this apoptosome, and the whole purpose was cleavage and activation of that initiator cascade. Yes?
So those pores are like channels?
They're holes. They're just holes. Yeah. Yeah.
Do they like, are they like
So you're asking how that pore is regulated? It's not regulated. It actually, the way it's regulated is by being there or not. So normally you'll have BH3, 1, 2, 3 proteins just in the membrane by themselves. But what they'll do is they'll oligomerize, so they'll bind to each other in a conformation that forms the pore. So when you have one protein just sticking in the membrane, it doesn't do anything. You need them to organize and actually form a pore. So it's not like a regulated channel or anything like that. It's only there when you have a stimulus, apoptotic stimulus. Good question. Any other questions? Okay, so this was from the quiz this weekend. You may have seen, you may have not. A defect in a BH123 protein, such as BAX. And so again, BH123 protein, going back to this, it's a whole class of proteins within this BCL2 family. And really, it means it has BH1, 2, and 3 domains. That's why it's called that. But anyways, a defect in a BH123 protein such as BAX will likely result in all of the following phenotypes, except, so who wants to actually take a minute to read through them and tell me which one of these is not going to happen if you have defective BH123. And I'll just remind you really quickly, and I'll put this back, what BH123 does. It sticks there, and then when you have your apoptotic stimulus, it forms this pore and allows cytochrome C to release. Okay. So take a minute. You have the answer? D. Disc will not assemble. Why did you choose D?
Because that's in the extrinsic pathway.
That is in the extrinsic pathway. Were you going to say the same thing? Yeah. So what you have to do to answer this is understand what the BH123 protein is, what it does, understand that it's part of the intrinsic pathway, and then understand the intrinsic pathway, and then go through each one and figure out which one doesn't belong, which is really just the one extrinsic pathway. Good. Any questions about that? Okay. So this, of course, is regulated. You have to think to yourself, well, what's keeping this from what's keeping these inactive BH123 proteins inactive? And so there are all these other proteins that are involved in this BCL2 family. So if there's no apoptotic stimuli, then the anti-apoptotic BCL2 proteins inhibit BH123 proteins from forming that pore. So they're just hanging out somehow, inhibiting oligomerization of the BH123 protein. And when you get activation of the intrinsic pathway, then BH3 only proteins, so there are these other proteins that are going to inactivate the BCL2 proteins. So you inhibit the inhibitor, which allows the BH123 protein to oligomerize, form the pore, and cytochrome C to be released. Right? So you'll have an inhibitor normally, and then you inhibit the inhibitor once apoptosis is activated. Okay? Give me a minute. Okay. So then there's this other thing. There are these inhibitors of apoptosis also, because sometimes you might get spontaneous activation of procaspases, and that would be really bad, right? You don't want just like, oops, apoptosis. No. So there are IAPs, and they're inhibitors of apoptosis, and what they do is they block caspases from being activated. And then so when you have apoptosis, so remember when you've got your BH123 proteins and they oligomerize, they form a pore, and you have your cytochrome C coming out, there are other proteins that live in that intermembrane space that also come out, and some of them are anti-IAPs, and they'll come and bind and block the IAPs, and that will allow apoptosis to turn on. Because your first question here should have been, wait a minute, so how does apoptosis ever happen? If there are these inhibitors binding to all the activated caspases, and that's because inside here you've got your anti-IAPs, and they come out and they bind and they inhibit, and that allows caspases to be activated. Any questions? Yeah.
So if you have your your apoptotic stimulus, even if you're stimulating apoptosis and you go through and the apoptosis stimulus is inhibiting the inhibitor and you release the cytochrome C, you're saying if the what are these things called? If IAPs are available, then they're gonna inhibit the capsid. So
No, well, so actually, so let me go back just to go over that. Right, so normally you have, you'll have your cytochrome C release, and that will result in activation of your caspases. These IAPs exist for spontaneous activation, so that's in the absence of apoptotic stimulus. You might just get spontaneous activation, so unregulated activation of caspases, and that's really dangerous. And that's why these IAPs exist, to prevent them from being active. But when you have an apoptotic stimulus, then not only is cytochrome C released, but also the anti-IAPs. And that will allow for apoptosis to happen, because you're then going to inhibit your IAPs. Okay. Make sense? Yes. Okay. Any other questions? Good. Okay. This is mostly it. Okay. So I mentioned survival factors before. These are these extracellular signaling molecules that inhibit apoptosis. And so there can be competition for survival factors. For example, in nerve cells, you've got your nerve cells and then you've got your target cells. And so your target cell will release survival factors, and that will allow that nerve cell to survive. But the different nerve cells are competing, so the cells that don't receive survival factors will die. So it's kind of like thinking reverse to what you would think. Rather than stimulating apoptosis by not receiving the survival signal through the survival factor, then you won't survive. So you'll actually you'll have apoptosis. So you can see here you'll have you go from 6 nerve cells, and they're all competing for these survival factors, and then only these might get it, and so half of them will die. And so that's one way to act. That's one kind of stimulus that would activate the intrinsic pathway, a lack of survival factors. You know what? I thought I deleted this one. I didn't want to confuse you guys, so we're going to skip this one. So the goal here, no survival factor equals apoptosis. So survival factors allow for survival, which would make sense because they're called survival factors. Okay, so I'm going to finish with a practice problem that you guys can work on together. And we'll go over this, and then that will be that. Okay, so you are studying the induction of apoptosis via the intrinsic pathway. You treat cells with UV, because remember, DNA damage can cause apoptosis. You treat cells, you guys are getting restless, you're ready to go. You treat cells with UV, not yet. I'm going to make you sit here until 4:45. No. You treat cells with UV light, and most cells undergo apoptosis within 24 hours. Cytochrome C release can be detected as early as 6 hours post UV treatment and can be detected for up to 10 hours longer. To figure out whether individual cells are releasing cytochrome C over a long time period, or whether different cells are triggered to release, that wording is horrible. To figure out whether, whether individual cells are releasing cytochrome C over a long time, or whether different cells are triggered at different points to release cytochrome C, you use GFP tag cytochrome C and follow it over a period of 24 hours post UV radiation. Which model does this data support? So you've got 2 cells you're looking at, and they're outlined. A cell at 10 hours and 9 minutes through 10 hours and 15 minutes, and a cell at 17 hours and 10 minutes to 17 hours and 18 minutes. And cytochrome C, if it's inside the mitochondria, you'll have this kind of punctate spotted staining because it's in the mitochondria. And when it's released, it should be diffused throughout the cell. So stop, talk to your friend, evaluate the problem, and then we'll go over it. Okay? If you dare. So what you want to know from this is, are single cells releasing cytochrome C over a 10-hour period, or are different cells being triggered at different times? That's what you're trying to figure out. Number one or number two, based on this data. So, you have a whole population of cells. Treat them with UV. Just use a pump. You can look it up, your whole population. If you don't know if over a 10-hour period of time one cell is really releasing, or whether one cell will get activated and another cell will get activated, another cell will get activated. I don't want to say that.
Actually, that's not yeah, it's just not
Yeah, but this means 10
All you do is
hours.
you have in
My sister, this means 17.
Seems like you should know that. You see that? I would start So the more more
Okay, guys, so tell me. So I know it was kind of confusing how the wording was there, so sorry about that. But the real question is, are single cells releasing cytochrome C over 10 plus hours, or are different cells being triggered at different times? What do you guys think? So I'm hearing lots of twos. Who wants to tell me why, how they came to that conclusion? Yes.
So I don't know if I was thinking about this the right way, but that's a matter of minutes, what we're looking at in A and B. So if at 10 minute,
Well, actually, that's
is it?
hours.
Oh,
This is 10
hours.
hours, not 17 hours.
Okay, well, then I was wrong anyway. So I thought it was minutes, so I thought if they were releasing everything in minutes, then it would have to be 2 because it's shorter than 10 hours, but I was wrong.
Alright, 10 and 17 are hours. What's 10, 9 and 10 and 10?
10 hours, 9 minutes. 10 hours, 15 minutes.
I think that's what she meant by minutes. Like once the slide at 9 is lit up and then 9 to slide at 15 is not like this one at the bottom 10 and 18 lit up and not.
Oh, from here to
That's
here?
why she's talking about
Oh,
Yeah.
different
okay.
times because within minutes is not lit up.
So you're saying it's going from non-apoptotic to apoptotic in minutes?
Yes.
Okay.
I don't know if that's right.
Well, I mean, it is, because you're not apoptotic here, you are apoptotic here. Okay, yes.
I was thinking it was different cells because the first was detected in 10 hours and then it went through apoptosis, but the other cell was detected until 17 hours. So
Okay.
then it didn't go through apoptosis.
Right, so the first cell was detected at around 10 hours, and the second one didn't go through apoptosis until 17 hours.
Because the cells around it too. The cells around it that aren't highlighted or aren't apoptotic at all. Like you can see in the
Right, right.
6 minutes that happen between the first two, the other cells around it are still.
Right, so you can tell by looking at the cells around it, and someone else said that too. Right, so the fact that this is not apoptotic, but this one already is, tells you a lot right there. Right, and yes, and then the time difference between 10 hours and 17 hours, the fact that that takes that long. Anyone else? Okay. Well, I am going to let you guys go super early unless anyone has a question. Don't run away. I'm gonna try to post this.